Hello, everyone. This is Steve Eth here, coming to you today to talk about two changes that have been made recently to the breast cancer cell line knowledge base. One is a change in how the Oncogene Signatures tool is set up and is now working. And in addition, I want to show you some additional pages I've recently added to the website so that all breast cancer cell lines now have their individual pages to make it easier to get information quickly for any one of the cell lines. I'm going to show you these two things today. Hopefully this will just take a few minutes and you'll find them helpful. So I will start by sharing my screen and we'll get this rolling. Okay, so I want to start here at the Oncogene Signatures tool page, and we'll scroll down here a little bit. And basically what you'll see when you go to the Oncogene Signatures tool page is in the original version of this, and in the version that's discussed in the paper, there were three tabs here. There was overall oncogene signature, candidate oncogenes, and then functional oncogenes. What I've decided to do is combine the functional and candidate oncogenes into a single tab to make it easier. And if you might recall, the difference between the so-called functional oncogenes and the candidate oncogenes was whether or not the gene was a hit in the functional screen. So the candidate oncogenes basically takes all of the known or candidate oncogenes from the OncoKB website, that 500 or so genes that are thought to play a role in cancer. And basically, with any of those genes have an alteration in any cell line, they appear in this table. And so the only genes that appear in this table are those that are considered to be bona fide candidate oncogenes. And what I've done now is I've just put them all in a single tab, and the ones that were hits in either the CRISPR screen or the shRNA screen are just now highlighted. And so you can now, at a glance, see all of the genes that are considered candidate oncogenes that have some genomic alteration, either a DNA amplification, as shown here with high-level overexpression for ERB2 in the AU565, or this Aurora kinase amplification, CDK12, or you can also see point mutations as well. And you can see the genes that were not only genomically altered, but were also hits in the functional screen. And so, that's basically the first change. I think this basically simplifies the whole thing instead of having two different tabs. Again, in the original version, the only genes that would have been in the functional oncogene signature are the ones highlighted in yellow. I didn't see any reason to really have those separate. You can now see all the candidate oncogenes for which there is a genomic alteration. And at a glance, you can see the ones that were screen hits, and you can tell if they were a screen hit in the CRISPR screen or the shRNA screen or both. So I think that really now just simplifies that information. And you can do that for all of the cell lines that are here in the database and in the website. You have all this information now the same way. So hopefully, again, that simplifies things. So the next thing I want to take some time to show you is something brand new on the website. This page of individual cell line summary. So again, for those of you who started off with this website because of the use of the some breast cancer cell lines, you already know that each cell line here has its own individual page where lots of information is sort of centered in those pages. So I wanted to try and do something similar for all of the other breast cancer cell lines that we now have in our database. And in particular, I wanted to be sure that everyone who with an interest in any given cell line could know very quickly, the age of the patient, the type of breast cancer, whether the line was derived from a primary tumor, a metastatic site, and so forth, and then have links to other resources for each of those cell lines, and then provide a summary, again, of oncogene signatures and drug sensitivities associated with each cell line. So what we've got now, if you click on the individual cell lines summary page, now you can see, and I have to tell you, this is still under construction, but I got it to a point where I wanted to begin to tell people about it. So each individual cell line here now has its own page, and I've started to add the keywords associated with some of the cell lines. So again, you can tell at a glance, if you're interested in the MDA MB415, this is an ER positive HER2 negative cell line derived from a pleural effusion. As I continue to work on this, all of the cell lines will have these sort of keywords UACC812 is an ER positive, HER2 positive, derived from a primary tumor, and so forth. So we'll have keywords for all of the cell lines in just a few more days. So stay tuned for that. So the idea basically is to be able to click on any one of the cell lines here, and now it has its own page. And what I've done in each of these pages is give you a little bit of information about the cell line. So BT549 is a triple negative cell line of the Claudin low subtype, derived from a ductal breast carcinoma of a 72-year-old woman. Now, so again, I think that's important information. And now I've got some links here, right? So the SCR profile, along with other data and references pertinent to the cell line, can be found here at expacy.org or Cosmic or DepMap portal. So what you can do is, if you want additional information, you can just click on any one of these, and you'll get the expacy page for BT549, or if you really want some in-depth genomic information, you click on that link and you're going to get the Cosmic page with all the stuff that that comes with Cosmic. And of course, you have a link to DepMap portal, and you'll come to the DepMap portal page for 549. So just direct link outs to these additional pages where you can find any kind of information you could possibly want. And we have this now for the cell lines. So then what I've also done is I've just now summarized the Oncogene Signature, which you could get from the Oncogene Signature tool for this cell line. But now I've just summarized it here and provided a little blurb about it, a little write-up on each of these. So, you know, this is a triple negative cell line, so the Oncogene Signature is fairly simple: P53 mutation, P10 mutation, and a little bit of additional information. But then what I've also done, and again, this is still a work in progress, is I want to link the Oncogene Signature to druggable targets. And again, I write some commentary about the drug sensitivities. Now, again, these drug sensitivities that I've put here are taken directly from the functional drug sensitivity tab or page here that we have in the data mining tool. So we have this breast cancer cell line druggable signature tool, and so all of the stuff that I've put here is just taken directly from that, but just sort of boiled down. So the functional druggable signature tool has got lots of information, and it's a good place to sort of play around and look for things. But what I've tried to do in this druggable targets table is basically focus on those drug sensitivities that connect directly to the essentiality of that gene, where we have both good gene essentiality and high drug sensitivity associated with each gene and each target. And so these are the things now that are in the individual breast cancer cell line table. We'll look at another one quickly here. AU565 is an interesting one. It's because it's an ERBB2 amplified breast cancer cell line. And again, we talk about that. And interestingly, and I note this for both, AU565 is derived from the same patient as the more famous SKBR3 cell line. HER2 amplification, P53 mutation, derived from a pleural effusion of a patient diagnosed with breast cancer. And again, the link outs to expacy, Cosmic, and DepMap portal. And so once again, now here are here's the Oncogene Signature showing the gene amplification of ERBB2 and its overexpression and so forth. And now, again, looking at the best druggable targets for this particular cell line, and highlighted here in red, here's ERBB2, all of the ERBB2 targeted drugs, and again, the scores, the CRISPR and shRNA score, but now you have the Z-scores indicating drug sensitivity. And so again, what I've tried to do in these tables is boil down all of that information from the functional druggable signature tool to only show you where there are genes and drugs where there's a connection between essentiality and good drug sensitivity. Because as we'll talk about in another paper that I'm working on, we think that when you have a good connection between gene essentiality and drug sensitivity, that really defines what a good druggable target is. Excuse me, particularly at the genomic level. So again, the idea here is to have an ability to look at any individual breast cancer cell line rather than using one of the tools and scrolling down to that cell line, but try and have all the information sort of in one place. And again, you can sort of use that as a starting point. So I'll just show you one more: HCC 1500, a HER2 negative luminal breast cancer cell line that's also estrogen receptor positive. Now you can see the estrogen receptor data here, see that it's highly overexpressed. It was a strong hit in the screen. The cell line has a cyclin D1 amplification, and KRAS was also a hit in the screen, and so forth. And again, a little description about the patient from which it came. So hopefully you find these additions and these changes to the website helpful, and make it a little bit easier to sort of use some of the data mining tools that we've built. Again, I think it's very important for people working with cell lines to know where the cell line came from, to know if it was a primary tumor or a metastasis, to know something about the patient, the ethnicity, the age. All of that, I think, is really important. And as I said, over the next few days, I'll be adding these keywords to the rest of the cell lines. Basically, it just occurred to me to do this the other day as I was sort of finishing this up. And I think the keywords are actually really important. ZR751 from an ER positive malignant ascites. It's very interesting. This is a cell line with the AP11 amplicon and a P10 mutation. And it's really important to know this stuff when you're working with these cell lines. So again, I'll be adding all of these keywords. So please play with this some. Take a look at all of this. Let me know if you have any questions. I'll be happy to chat with any one of you directly by email, by Zoom, whatever it takes. And I appreciate any feedback. And if you think there are some things you'd like to see on here, I'd certainly appreciate hearing that as well. So thanks for taking a few minutes today to watch this. I'll post this video on my YouTube page, as well as on the Breast Cancer Cell Line Knowledge Base page. And I'll be sending out a blast email here momentarily to tell you about this new video and about these new features. So thanks a lot, and I'll see you next time. Bye for now.